This is the fifth lecture, and which is the second part of the DNA sequencing technology. And the first lecture about DNA sequencing technology, we basically trying to overview a lot of basic theories or biological applications, and trying to tell you how great this technology is and things are like that. Present some of the successful stories, including finding the disease that caused the gene that caused the disease, or for the clinical applications. And today we are going to, and so the last lecture was reasonably exciting because it's more on the biological application. It gives you good stories and things like that. But today the lecture will be much less enjoyable. We will come back and to really get your hands dirty and to see how are we going to analyze the data. So how to make those good stories, which is not necessarily simple. The topic here is bioinformatics and statistical topics. So a couple of review slides of last week. We first discussed what are the genetic variations. And there are different types, and the most common ones are the SNPs, single nucleotide polymorphism. It probably happens for every 1/300 nucleotides. And we also know that those SNPs, and they can be inherited. And to qualify us to be a SNP, one important thing is it has to be represented in over 1% of the population. Okay, otherwise, definition can be loose, but people generally don't. If there's one single nucleotide variation only presenting in your genome, that's called a mutation. It's not really SNP. It has to be population-based. And there's a lot of short insertion deletions are pretty interesting as well. And many different types of repetitive elements, including the microsatellites, minisatellites, and repeated genes. These are basically divided by characterized based on the length of the repeated elements. Some of them are really short. They are called microsatellites, and others are bigger. And the last type of genetic variation we discussed a little bit is called large structure variations, including large insertion deletions, duplications, inversions, copy number variations, and there are many different types as well. In the end of this lecture, and we will have a couple of slides to talk about the methodologies being used to identify these large structural variations. But most of our topic will still focus on the SNPs, which are the most common genetic variants. Before we had next generation sequencing technology, we know that technology really allow us to study genetics of disease in the whole genome level. Very early it was based on the microsatellite markers. So there's only a couple of hundred markers across the entire genome, while the SNP arrays become more and more popular and more capable, and there are 1 million or 2 million SNP arrays, meaning there's a couple million SNP markers on one chip. So your resolution becomes much higher. And for this part, its most analysis will be the linkage type of analysis, and further becomes association type of analysis. We also spend quite a bit of time to talk about the GWAS, which is Genome-Wide Association Studies. These are, if we talk about the DNA sequencing, these are something you have to talk about, because DNA sequencing is kind of have the general notion that will replace the GWAS in a couple of years. So what the GWAS can not do and the next generation sequencing can do. So the underlying hypothesis for the GWAS is a common disease, common variants. Here you can see this figure, and this X here is how common the variants are. So this is a very rare and becomes a little bit more common. And the Y here is the effect size, so how important that variants are. So some of the variants, they can be very rare, happens in 1 out of 1,000 people, for example. But once you have it, you're sick. And those are pretty important variants as well. So the effect size will become bigger for those. In general, that GWAS is working on this part, which is more on the common variants. and the common disease. But these common variants, in general, their effect size are smaller. We are hoping that we always get things like that, but those are the diseases that people already studied. So those are lower fruit size has already been picked. And for the family-based linkage type of analysis, and they are pretty capable of identifying this type of rare alleles, but it can be pretty rare. For the next generation sequencing, and we are hoping that we can go a little bit beyond this spectrum and looking at rare allele with there's a kind of rare allele, but with intermediate effect size. Hopefully, we can get to that as well. But there's a lot of, and also there's a lot of limitations of GWAS study. And the first one is it do not capture the rare variants. So we know that because everything that you print on those SNP arrays, which is used in the GWAS, they are pretty much known known to the general population. They are common. And the second one is its lack of functional information, meaning that most of the SNPs on these GWAS arrays, they are so-called tag SNPs. So they just a flagging. It's a flag of a closed region. So if you identify one variant, one SNP is associated to the disease, that doesn't really mean that variant is causing the disease. So it's not, doesn't have the functional information. It's just a flag. Say something is wrong around this area, and we need to go deeper to study that. And we also know there's a lot of missing heritabilities, and there's a very nice paper about that. There's probably, it's a combination of multiple reasons for that. It can be environmental factors, can be gene-gene interaction, can be variants, rare variants, and it can be multiple things. So there's a lot of limitations of GWAS studies. Next generation sequencing technology really help us dramatically, at least in the beginning years, to understand the genetics of complex diseases. Well, we talk about 3 examples. The first one, they all published in Nature Genetics maybe in 2010, 2011. And the first one is the disease itself is not important, but the papers, the first one represent the example of whole genome sequencing. After genome-wide association study and get whole genome sequencing, and they identify some meaningful biological results. The second one is Miller syndrome, which use exon sequencing, which is a very good study as well. And the third one is a study that used the pooled sequencing. So if you're interested, go back to the papers to look for details. And we also talk about, so the previous slide is really trying to understand the genetics of complex disease. And for the future, next generation sequencing can be used in personalized genome sequencing. We also talk about 2 examples. The first one is published in Lancet last year, and that is really a clinical assessment of a 40-year-old man who present with a family history of chronic artery disease and sudden death. But that man is generally healthy. That's a healthy guy. And he went to the hospital, did a lot of clinical reports like blood pressure and blood sugar and all those things, which is very regular. But once he got his next personal genome sequence done, we can really further to refine those clinical reports and to predict at which age, or maybe not which age, but whether this person has a risk for certain disease. In combination of the personal genome sequencing data together with the clinical reports. That's very important information. And another very, very exciting story of this little boy, and he had a disease which nobody knows what is causing that, but after exon sequencing, and they did find the variation that caused this case to be sick, and they even figure out a cure for him, which they did cord blood transplantation. But this kid is really a lucky story. So you identify, you sequence one individual, identify the gene, it's important, and you have a cure for that, which is lucky. But hopefully this represents the trend for the future clinical practice. We also mentioned that after the story of this, many hospitals in the New England area, they already had launched a pilot that if patients want to do that, they can start to think of the exon sequencing or even whole genome sequencing. And I think somebody sent me an email during last week after a lecture that NIH is having a similar pilot project as well. So it becomes a trend. So it represents the future of clinical practice. And the last point that from the previous lecture is we had a couple of slides to talk about experimental considerations. The first one is whole genome sequencing. I mean, this is really on the DNA sequencing side. The first one is whole genome sequencing. The major concern is the dollar amount. So we don't have that much money to sequence many individuals to figure out what is causing the disease. And also one very good solution will be the target sequencing. And we briefly mentioned the capture region selections. It can be from other studies. It can be GWAS or linkage studies say this region is important. Or if we can only focus on exon regions, which are likely to represent more biological signals than the rest of the genome. And we mentioned a little bit about capture technologies, which include the capture array and the capture in solution. So if you recall those beautiful slides. And the last one is the pooled sequencing. So those are the basic biological part of next generation sequencing technology. But today, what we are going to go into is really the data analysis part. So there are a lot of statistical and bioinformatics theories behind the different ways of calculating things. So we often have heard from people that the most recent was on the train. So Consik told me that somebody in the group did exon sequencing and they did alignment, they called the variance, and did annotation, and it's very simple, and they just got the list. Okay, and if you talk to somebody who really did the analysis, like Janice and Hai, and they will tell you that's not that simple. They struggled months after months and still sometimes have the feeling that go back to ground zero. And so the point here is once you finish the analysis, once you finish the experiment, when somebody did analysis for you, for example, did alignment and did variant call, you want to ask them, what is the algorithm did you use? Has these things been considered? And if this is the approach you use to call the variant, what is the assumption of that approach? Is that fit our experimental design or things like that? So before you ask those questions, you probably need to know a little bit about different components. That's the purpose of this lecture. So we got the raw data. Basically, that's a lot of ACGTs and together with quality numbers and in the server. So we know that the file sizes are pretty big. Sometimes it can be 10 gig of one file. And Hai just told me that today he was trying to transfer some data to one of the collaborators, and the hard drive they brought in was in FAT32. So they cannot really manage any file that larger than 4 gigabits. That's how severe the problem is, right? So even you bring in the hard drive, you have to bring in the right format. So because of the data. But those data, as good as they are not very useful if you don't analyze them. There's such a huge amount of information. So we know that from that we have this BAM file, which is after the alignment. So you got the individual sequence arrays. You want to align them back to the genome. And we also talked about the alignment algorithm last time. And after BAM file, and you want to identify the variant. So you want to know that how many variants are in these individuals, and then you start to make story of those. So what we are trying to get at today is the first two topics are on alignment side still. The first one is the sequencing mappability issue, and this is a very important issue that we all need to consider. So because our human genome are so competitive, and so repetitive, not competitive, so repetitive that there's a lot of regions that we've got to sequence, and it can be mapped to multiple regions, and you don't really want to trust those part of signals. And also, we are going to spend a lot of effort talk about realigned algorithm. And so refined alignment, this is very important. I can show you the example of that. And we also want to talk about the quality recalibration. So the raw data, so remember we talk about, when you talk about the quality, there are 3 level things. The quality for the individual base, the quality for the mapping, and the quality for the variant. So there are some new algorithms trying to recalibrate the quality for the raw base and the quality for the variant cost, which are extremely important. And most people just don't even know there's such thing like that. And once you talk to people who did that, and so those are the questions probably not even cross your mind, and now it is. So you want to ask those questions, so they know that they cannot fool you, right? And we will also talk about variance identification, and which is variant call part from the alignment, the aligned file to the variant call files. And this will also have many different potentials, including the diploid genome, multiple individuals, pooled DNA sequencing, and cancer genome. They are all different. So the analysis itself is different. So people often say, we call the variant use SAM tools. All right, so SAM tools is probably already installed in your query account, your account, which is fine. But sometimes you can use it, others you cannot. So we want to know that what is the assumption for that different approach. And I will spend a few minutes talk about identify structure variations, which is some topic that I'm not expert of. I'm just still learning, and we haven't done anything on that. But by reading a couple papers, there's a very active research topics in those as well. So let's start from the alignment part and talk about the sequence mappability. So have this mappability issue ever crossed your mind? Have you heard about that or think about that? Okay, so let's see, for human genome, for one sequence fragment, what is the minimum length can be uniquely mapped back to the human genome? Who can answer this question? What is, if we got a DNA sequence, I don't know how many base pair, it can be 10 base pair, it can be 100 base pair, what is the minimum length and can be uniquely mapped back to the human genome? I know 2 nucleotide is not going to make it, right? Every 16 base pair will have one. Probably not 3. But is that 100 or 200? Who have think of this problem? Have you make any calculations before? So a very simple math is the size of a human genome is 3 billion letters, okay? And each nucleotide have 4 choices, A, C, G, and T. And if, in theory, okay, this is not going to be accurate. We know it's not. In theory, if we have an N nucleotide read, and the full power N larger than this, N is about 15.7. Okay, that means in theory, reads with 16 base pair or more can be uniquely mapped back to the human genome. Does that make sense? Okay, that's just in theory, because there's so many things are still repetitive. But in theory, that is the length required. So that's why that when you guys design primers, I know a lot of biologists in this room, when you design primers and to do the PCR reaction, you want them to be 16, 17 at least to 23, 24, and that range for temperature considerations. Is that right? But you still, once you design the primers, 16, 17, 18 probably is okay, but you want to still try to see whether it falls into the repetitive elements. But the point here is, in theory, reads with 16 base or more can be uniquely mapped back to the human genome. Okay, so we have this data. Now we are excited. We say, oh, we are having 50 base pair. We are in good shape. So there's no problem at all. It's not necessarily true. And the reason for that is half of the human genome is repetitive DNA. Okay, as we have 3 billion letters, and 1.5 billion are junks. Well, not repetitive. I don't try to say that repetitive are all junks, but a lot of them we don't know how to analyze those. So the repetitive DNA elements are found in multiple copies, in some cases thousands of copies. And there's different types of repeats, and there are some type called tandem repeats. Those are mini satellites and microsatellites. We already talked about that. And there's interspersed repeats, including DNA transposons, LTRs, non-LTRs, signs, lines, and alus, and those kind of things. If you don't know what they are, that's perfectly okay. But the point is that they are very repetitive. So this is one of the figure from actually Wikipedia. This is trying to, in the Genome-Wide, trying to identify the ALU elements. So all the green regions are enriched for the ALU signals. ALU is, I think, is I don't remember how many base pairs. It's a couple hundred base pair, and I think, and it's so you can see all the green regions. Some of the chromosomes, they are completely ALU, it seems. So it's very, very competitive. But the take-home message for here is half of our genome are repetitive elements. So now come back to that question. So 16 base pair, well, in theory it's okay, but not quite. But how many base pairs is enough, right? And so here is one of the study that I don't know whether it's published or not. I got this from internet somewhere. But the way I interpret this is the length of the nucleotides, and this is the percentage of genome that can be uniquely aligned based on the length of nucleotide that we are providing. Okay. For example, if we provide a couple of nucleotides, it's not going to make it, and nothing will map. But if we got 100 base pair rays, okay, for Illumina technology, it's very typical, 100 nucleotide base. And for different species, you can see that most of them reach the plateau, which means that going further will not improve your mappability, okay? And here is for human. Which color is human? Human is this one. I believe it's this one. So it's about a little bit less than 80% of the human regions can be uniquely mapped. Okay, and I mean the rates can be uniquely mapped back to the human genome if we are getting to about 100 base pair rates. Okay. For some of the species, they become shorter, around 20 will be fine. But for humans, it seems that it's still increasing, but it's not increasing much after we are getting to 50 nucleotide rays. Okay? So this is a piece of important information that you need to keep in mind. So mappability, or other people call it uniqueness of reference genome. So we are talking about the different part of genome are so repetitive. But point is, which part of genome is repetitive? Is there any way that we can find that out? So actually there is. There's people already, some smart people already did a calculation, calculated this part of genome, whether it can be, it's very similar to other parts of genome as well. So it's available for download at UCSC Genome Browser and display how uniquely K-mer sequence aligned to a region of genome. And K is 24 to up to 100. Okay, and here is the figure from the Genome Browser. So you can see that for the 24 nucleotide, and I assume that the larger, this is definitions of the y-axis here. I'm not going to go into that, but the point here is the larger the value is, the higher the mappability for that region is. So if you got to the top, that means that part can be uniquely mapped. Does that make sense? So you can see that for the 24 nucleotides, there's some part that it cannot be really mapped. Even for this part, and the mappability start to drop. But once you got to 100 nucleotides, and the mappability is really, really good across the entire region, except for a couple of regions that no matter how you try it, it's probably going to fix it. Okay? But when do we use this mappability information? This is good information. We know that for certain part of the genome, we are needing longer reads or things like that, but we are pretty much fixed at 100 base pair or things like that. But how we use this information? Well, actually, when identifying genetic variants, so this consider this information is really, really important. Think about this. If there's one read, okay, it can be mapped to both two places. Okay? If you don't consider the mappability here, and you just you're blind, you don't know this is happening, and then so if this region is a homozygous, this is both two alleles, parent are the same. So here is a C, and this part is very repetitive. It's very similar to here, but just one nucleotide difference here. It's G. Okay, so this is a two homozygous part in this repetitive region. But when you read the data, when you misalign these reads, come here, and you probably will find that this region is a heterozygous. Okay, and this will be the wrong conclusion. This kind of artifacts is completely caused by the mappability issue, because these reads can be mapped to multiple places. Okay, this read mapped to here, some reads mapped to here, probably is misaligned. Does that make sense? Okay, so this is something we really, really need to consider, be extra precaution after identifying these variants. And also for quantitative measurements when comparing two or more genetic regions. So let me explain what that means. So for the RNA sequencing experiment, we know that there's a gene. There's two genes here. Okay, there's this gene and this gene. Their expression level is about the same. Okay, and when we're trying to evaluate how much this gene express, how much this gene express, your conclusion probably is that they are expressed equal amount. I think, which is fair. Okay. However, if for this particular region, that it's somehow not identical but similar to a region that in another part of the genome, and if there are some of the sequence reads that can be mapped to that as well, if that part is expressed as well, and your end result is you probably will misalign these regions back to here, and then you will think this gene expressed twice as much. Okay, that is the wrong conclusion. So that's what I mean. For quantitative measurements, when comparing two or more genetic regions, for example, RNA-seq, and mappability is something you really need to consider. Okay? The second example, and we will talk about it towards the end of this lecture, is the copy number variation. So we know that for this one, there's about the same copy. Okay, if there's something that in other part of the genome is the same issue, and your signal is, because of misalignment, your signal is double the signal in this red region compared to the blue one, your conclusion might be this is a copy number. It's a duplication here. Okay, that is probably a wrong conclusion. That is completely come from this artifacts of misalignment. So the third type of application is when you design capture arrays. Okay, we know that we talked about last time. You can design the capture arrays in the genetic regions you're interested in, or you can capture exons. But when you design these capture arrays, so for example, this is the region that you're trying to target, and most likely your experimental design will be targeting things like this, right? Tell the entire region and the design probes targeted. But when you have a region that within the target, whether there's a repetitive region there, okay? If your PCR-based capture, and within the target region, if there's a repetitive elements, meaning that this region and this region, the sequence are very similar, and you probably will get a pretty wrong conclusion as well, because when you do the alignment, both these two parts will be maxed up. We don't really know that for one read, whether it's come from this part or that part. Okay? We had that issue when we work on Howard Edinburgh's data on the ADH regions, because the ADH, there's ADH 1 to 7, and the 1A, 1B, 1C, their genetic, is that correct? 90% identity for the sequences. But once we design the captures, PCR-based assay, and we didn't consider that, and that created a lot of trouble for us to do the analysis, because there's a lot of variations just simply because of the misalignment. If we if we taught this lecture earlier and I thought this through, probably will design experiment a little differently. But those are something really need to be considered. And for the hybridization-based capture, like capture in solution or capture arrays, and you are not only considering within target regions, you also need to consider between outside the target regions as well. Because when you target, you are trying to target this part. However, if this region and this region are too similar to each other, when you do the capture assays, and this part of DNA hopefully will be put down, but this part will be put down as well. Okay? And then once you do you call the variant and things like that, and eventually it mess up with things. So you don't know exactly that fragment is come from here or there. Okay? So far, I really haven't seen a really good program to help us to solve this problem. And so before you design the exon arrays or any type of capture arrays or capturing solutions, and trying to figure out a region that not really repetitive with any other nearby regions. And if we have a program like that, that will be very valuable, I think. And the next point I want to deliver about mappability is you need to probably generate your own mappability tracks. So a couple of slides ago, I showed you the UCSC genome browser mappability track. So you can see that it's a 24 base pair, 36 base pair to 100 base pair. The mappability are really different. But actually, there's some other factors can determine the mappability issue as well. And the most important one will be the alignment algorithm. So some alignment algorithms are good at identify distinguish small differences between this region and that region. Others are not. So it's alignment algorithm can be a consideration. And the regions where mapping occurred is important region as well. So for the previous slides, we know for the PCR-based assay or capture-based, hybridization-based assay, your consideration of the repetitive is different as well. So we need to really think through. And the biochemistry assay is different. Regions can be slightly different. Okay? And as I mentioned, that so far I really don't see a general strategy that trying to help you to identify the mappability and before and you do the experimental design, and after the experiment, do the analysis. I think this is still very important research area that we need to go into. Maybe a simple program that can help us to generate our own mappability track based on the strategies we are using will be really, really helpful. Does that make sense? So the mappability part, is that okay? Okay. Now we are going to move on to the refined alignment. Okay. So we have spent quite a bit of time in the second lecture talking about alignment algorithms. Start from Smith-Waterman and talk about hash table-based index algorithm. We'll talk about the prefix tree or surface tree. Talk about the BWT, and we were really painful going through those calculations. Remember that? But after all this, we still don't trust our alignment? And the answer is yes. There's a lot of regions that refined alignment will be really, really helpful. Okay, so let's look at the problem first. And the goal of refined alignment, so the sequence alignment, number of mismatch bases minimize number of mismatch bases minimized across one read. So this is actually not the goal of refinement alignment. This is the goal of our initial alignment. So we got the sequence reads. We want to map it back to the reference genome. Okay? And our goal is to look for this one read at a time. Not consider multiple reads, one read at a time, and trying to minimize the mismatches. So here is the reference, and this is the donor. So we're sequencing. There's a SNP here. There's a couple of deletions. And the goal bottom line is across one read, but the sequence for the refined alignment is the number of mismatching bases minimized across all the reads. Let's see why it's important. And what went wrong for the initial alignment? So a large percentage of regions require local realignment are due to the presence of insertion or deletion in the individual's genome with respect to the reference genome. Okay? And it's not very clear, but this figure that Hai gave me shows really well. So let's go through this. Here is, this is the coordinates, okay? And this is the reference genome. And this is our sample. So you can see there's a couple of insertions in this region, okay? But when we do the alignment, we don't know this. We don't have this, okay? We don't have this sample. This is totally blind. Okay, so when we do the alignment, we got 6 different reads here, and for the first read, so you can see that because I don't have this gap, this I don't know this, so you can see this AAAT, and it map back to the original reads, and with one nucleotide mismatch. So you can see this if I align this way. Is that right? So you see here's the reference. And this one has one nucleotide mismatch. Here, I didn't know that there's an insertion deletion here, so just ignore this part. Okay? And for the next read, actually becomes this. So there's the same thing, and there's 2 mismatches comparing to reference genome, because when you're really trying to align one individual read, you are trying to align one individual read, having one SNP is having much better score than having a big insertion inside, okay? And the same thing happens here. So you can see for this one, I ignore this part. There's 2 things, mismatch and one mismatch, which is still, the algorithm thinks it's still better than put in insertion here. But this read, so you can see that it's much better to have an insertion in here, and because everything else will be perfectly aligned. Okay, and so this, if the aligner prefers 1 or 2 mismatches over a full base pair insertion, that's what happens. Okay, so if in reality this, if I align this particular race, this AAAT, AAAT, AA, and if I put this in insertion here, that is probably give a much better performance. It's become a perfect match if I know there is a insertion in that region. So this is really tells us that when we do the initial alignment and we are trying to optimize the locations for one individual race, not to consider all the race as a whole. Okay? And such alignment artifacts result many base mismatching the reference near the misalignment part, so and which are mistaken as SNPs. Some of them will be mistaken as SNP. There's another thing, observation here is, so most of these you can see is towards the end of the race or the beginning of the race where misalignment happened. If this insertion happens inside the rate, that's probably less likely to happen. But anyways, this will give you really a lot of misalignment. So initial mapping treats each read independently. Even when some of the reads are correctly mapped with indel, reads covering the indel near just the starting ends are often misaligned. So for these ones. So local alignment serves to transform regions with misaligned due to indels into clean reads and containing a consensus indel suitable for standard variation discovery approach. There's the 2 steps. Okay, so basically the point here is the local realignment is trying to fix the previous problem. And there's 2 steps to do that. The first one is to identify small suspicious intervals which are likely to have a small insertion deletion there. And for those regions, we probably need the realignment. So we need to find that regions first. And the second step is to run the realigners over these intervals and to have a better results. So the first step, let me turn around to show you the result first, and then we come back to the methods. Okay, so you can see that this is one of example. We have observed if this is the initial alignment, there's no realignment happened. So each bar here is a read. So you can see in this region, we identify the 2 SNPs in this region. Very clear SNPs in this region. But there's a deletion here in the reference genome. But once we do the realignment, it becomes this. Both 2 SNPs gone. Okay? It's not something magic. We will go back to the mathematical equations, how they do that. But the point is this insertion, this deletion really helps to create these 2 SNPs which are not real. Okay? After you consider the insertion deletions and to do the realignment of a part of race, and those 2 things gone. There's another example here is also similar. So there's another region. This is all I believe is 1,000 genome data. And you can see that this is one region has insertion deletions, but the others really had, you identify 2 things and you think these are heterozygous variants. So these are pretty much look like that. But when you do the realignment, everything is gone. Okay? So this realignment really is helpful. Yeah?
Do you know how long these reads are?
I believe this is high-seq data, so it's 100 base pair reads. Yes. It probably will be less, but still have a lot of problems. And the point is, once you identify a couple of thousand SNPs and single nucleotide variants, and those are the ones you really look at that is really, if you have a few hundred are caused by these realignment issues, those are never going away unless you fix it. Okay, so are we clear about the problem itself? Not know the solution yet. So we can know, right? So let's go back to the equations and to look at how they do that. So we mentioned there's 2 steps to do it. The first step is to determine the suspicious intervals, identify those regions that's probably we need to realign. And the second step is to do the realignment. Okay, for the first step, to identify the regions that are more likely to be, there's misalignment there. So the first strategy is we want to identify the de novo indels in the initial alignments. Okay? If one or more reads contain an indel in those regions, and those are a red flag. Okay? If there's 100 reads or 50 reads, and there's 1 or 2 reads has an insertion deletion in those regions we need to be really careful. I mean, we may need to go to do the realignment for those regions. And if there's no indels identified in the initial alignment, but we still, if we already did a base call, so we know that which has ACGT, how many of those, and we want to search for the cluster of SNP calls. So if 2 SNPs or 3 SNPs, they are very close to each other, and those are probably a region you want to consider doing realignment. And if we haven't even done this base calls, we want to detect the clustered loci with high entropy, meaning that there's a lot of mismatches in those regions. We want to consider doing realignment on those. And also the third component, the category is for known indels in dbSNP, and for those regions, we certainly want to do the insertion deletions. But this is the general step 1, trying to identify the regions that we need to do the realignment. And the second step is to really do the local realignment. So there's also 2 steps in here. The first one is we want to construct all possible haplotypes by interrogating the reference genome, known gaps, and the de novo gaps by our alignment algorithm. And then the second step is to conduct a gapless alignment against all possible haplotypes and calculate the likelihood of each haplotype. Here's what that means. So you have the reference genome, okay? And if you have 1 or 2 rays that supported there's an insertion here, and you want use this as one of the candidate of a haplotype. Okay, so you create a new insertion, a new reference genome around this local area. Does that make sense? Okay, so usually only the reference genome are visible to us. We only know the reference genome. We don't have this. But if one or two rays presented an insertion here, we may consider to do, this is a reference and this is another possible reference, which is another alternative haplotype. And then for all the rays, we want to align to both two haplotypes with ungapped alignment. At this stage, I don't want to do gapped anymore because the gap has already been considered in this haplotype. Okay? And the equations to do that, so I'm just going through this. If you understand, you understand. If not, that's no big deal. But the point here is you want to calculate the likelihood of each of these two haplotypes. Sometimes you may have more than two haplotypes, okay? And to calculate the likelihood of this haplotype, and then you want to calculate this is the number of rays in this region, and how many rays, what's the likelihood of that, given that haplotype, how many rays are correct. And this likelihood is calculated here, which is a sum of number of mismatches in those regions, and actually these are just the quality measurements for each different mismatches, and this is a match and this is a mismatch, so you have different likelihood. So the bottom line is you have two potential haplotypes, and you want to align all these rays back to these two haplotypes, and you want to calculate the likelihood that both two whether this new haplotype is a possible solution as well. Okay? And a little bit of math on this. So the haplotype HI, which is the new haplotype, the ones that we see insertion there, that maximize this LHI is selected as the best alternative haplotype. Okay? And then we assign all the rays either to the original reference or to this reference with the best haplotype with this insertion there. And the rays are realigned if log, okay? So here is the thing. So I created this new haplotype in these local regions, but it better to improve the performance of the overall alignment. And the way to evaluate that is once I cause that much trouble, I better receive enough benefit, which is the rays are realigned if the log-odds ratio of the two haplotype model is better than a single reference haplotype by at least 5 log units. And at least that's the cutoff they use. If I achieve this goal, and I think we need to do the realignment for this region, there is a small insertion in this region. Okay? Does that make sense? Okay, we just go through this back again, just conceptually. Okay, so we already see that with the insertion deletions in those regions, if we are not careful, that it will create a lot of SNPs in the surrounding regions. So if we observe an insertion deletion, or if there's a known insertion deletion in that region, that is already a red flag for us. We probably need to do the realignment in those regions, and that is identify those regions. So once we do that, the way we do that is we use the reference genome as a possible haplotype, and then we use we create alternative haplotype by putting the insertion there, and then redo the alignment with all the rays using ungapped alignment, because at that stage, the gap is already taken care of. The insertion and deletion is already taken care of. And if I cause this trouble using this two haplotype model, I receive enough benefits, which is the performance is much, much better than using a single reference haplotype. And then I need to do this realignment. Okay? Is that conceptually okay? Okay. If you still don't understand, just keep in mind, around the insertion deletion regions, there are a lot of misalignment. Just do it again, okay? Use the algorithm. All right, so this is examples that we already went through. But there's some key information on this realignment algorithm. So comparing to the initial alignment, key difference is really minimizing mismatches for one reads versus for multiple reads. That is the key difference of the initial alignment and the realignment, okay? And there's a two-step procedure, identify regions and conduct it, and realignment process to do is first enumerate the potential hypotype candidates, conduct gap-list alignment on all hypotypes, and calculate likelihood for each haplotype. So which one is really better? But use with caution, even for the realignment. So just we really need to think through. And major assumption for this alignment is what? Is this algorithm operates on all reads across all individuals simultaneously, which ensures consistency in the inferred haplotypes among all individuals, which is good. But it really doesn't work on somatic SNPs or indel and indel callings. And because for those somatics are meaning for the cancer, right? For the cancer tissues, and there's a lot of cells that created during the cancer carcinogenesis, and they created those kind of insertion deletions, not on all the cells, only on a few cells. And if we do this, that really is not follow the assumptions of this realignment. And it also needs to have significant improvements to replace the initial alignment. So if we use this one haplotype model or two haplotype model has to be significantly better than the original one 5 times. And then you can really use it. But this is for the pooled sequencing experiment. If only a small proportion of individuals has that indel, it's not going to work. It's not going to receive enough benefits to overturn the previous using one haplotype model. And for the RNA sequencing experiment, if the low expressed allele contains the indel, low expressed allele meaning that if 2 alleles, one express this much, the other one express this much. If this much one has an indel there, it's not going to work. It's not going to fix it. So coming back to this is we really need to be careful about this. And so that's what I mean. So if you download the software package and they can really do the realignment for you, but you want to know what they are doing. And once you go through these mathematical equations, maybe you don't understand it, that's okay. But as long as you think through their assumptions and all these things, you want to make a decision whether you want to use it or not. We have so many experiments that can benefit from this, and we have so many experiments that this can really not help us at all. For example, our pooled sequencing experiment, for example, our RNA sequencing experiment. Okay? So we are about a 1 hour mark, and let's take a 5 minutes break.